Like submitted it, but I realized it said 7 days.
So I changed the deadline. Generally speaking, I try to hold true to my word that lab is due the next time we come to lab. So if we miss a lab for a snow due day or something like that, then it just moves forward to the next due date. That does mean that if you rushed to upload something, you can still submit a corrected file through Wednesday, because I'm not going to look at them until the due date. So you have that window.
So we can just hand in that graph.
You can totally just hand in the graph in paper. Yep, some people have already done that. I have them in my bag. Yep. Any combination of partially uploaded and partially paper, or all one or the other, is fine. Okay, so we're almost through chapter 1. I feel like we're kind of far into the semester to still be doing chapter 1. It's okay. In lab on Wednesday, we're going to expand some of our concept of chapter 1 by talking a little bit about the information life cycle and the scientific method as it relates to where science information comes from. So actually things are pretty well lined up now. I didn't like the previous schedule. I have to be honest, the snow day made me really happy in terms of pushing all of our labs forward, so that we're a little bit more in line with the lecture content. So we were talking a little bit about controlled experiments, and you guys had thrown out a bunch of things that we would want to control if we were going to do an experiment to test the effect of fertilizer on growth in plants, right? And some of the things that you threw out, if we were going to answer this question, does application of fertilizer affect plant growth? We would want to make sure that we used the same plants, same seed stock from the same source, grown in the same year, right? The same soil, and both in terms of what is in the soil, what the makeup of the soil is, but also the quantity or volume of the soil and the pot that it's housed in. The same amount of sunlight exposure, the number of hours per day, and the angle of sunlight should be the same. The same amount of water. All of those things would be the same. And the only thing that we're going to manipulate is that variable of fertilizer, right? And we could have 2 treatment groups. We could have some plants get fertilizer and other plants not get fertilizer, and just look, do we have fertilizer or not? Or we could have sort of a continuum of treatments. We could have no fertilizer, a little bit of fertilizer, a medium amount of fertilizer, and a whole lot of fertilizer, right? And we could scale that and test and compare those groups, okay? That's a very controlled situation. If we take these plants into a greenhouse and try to do this experiment, we can control all of those factors, right? But as soon as we remove that sort of artificial construct of the lab or the greenhouse, and we want to ask the same question in the natural environment, we can't assume that our entire field, if we're going to look at a cornfield or a vegetable field, that our entire field has the same soil or has the same nutrients in every part of that soil. Maybe a deer died in that field 2 years ago, and there's a hot spot of nutrients right there where that decomposition happened that's not anywhere else in the field, right? I had goats as a kid. I pitched my goat manure over the fence into the farm field. The corn was universally a foot taller in my pitch zone than it was anywhere else, right? If you've got some supplements, and you can't control those things in an experiment if you're in nature. So sometimes when we take things out of the lab, we have to accept that we can't as tightly control these variables as we might want to. And then we have to just observe the differences and try to take those into account in our mathematical modeling. Okay, if I wanted to look at the results of this experiment and see, does the application of fertilizer affect plant growth? How am I going to measure plant growth in this population? What could I record? Height? Like some measure of density or volume of the plant, like how many leaves per stalk, or how big are those leaves? Yep. Anything else? Rate of growth. How fast does it change? Color. Yeah. Color changes in plants are known indicators of nutrient stress. And so if we have some plants exhibiting various nutrient stress colors and others have a nice rich green, that's a good one. So how would you know something about the quality of the plant? Okay, does it look robust, right? Does the plant look healthy? A what? Amount of leaves, like total number of individual leaves. Yeah. All of those are things that we might measure. At the end of the experiment, we could also harvest the plant and we could look at its actual biomass. So we could dry the plant to remove the water and measure how many grams of leaves did we have, how many grams of stem, how many grams of roots, and just put it on a scale and measure that in a controlled experiment like this.
Actually did
Environmental science, yeah. You had different pHs and salt contents in the water. Yeah. Yep. It's a really straightforward experiment to do and gives you good answers, right? You might also look at reproduction. Does the plant flower? How many flowers does it produce? How many seeds does it produce? Because that would indicate that it has the resources available to do those things. Because all reproduction in all organisms is expensive. It takes resources. You have to have the energy available to do that. And so that would be another thing we could measure. Okay. So what I want to do now is walk through the scientific method in this oversimplified example. Okay. So I know that most of you don't even use flashlights anymore. I'm going to have to update this soon because you just get out your phone, right? Turn on the flashlight on your phone. But our individual here has a flashlight and she goes to use it and she notices that the light doesn't come on. Okay. So our observation is that the flashlight doesn't work. What's our question? Why doesn't my flashlight work, right? Now, there are multiple potential reasons why the flashlight doesn't work, but what is your first guess?
batteries.
Dead batteries, right? Dead batteries are kind of always the first guess. So my hypothesis is an answer to my research question. If my research question is why doesn't the flashlight work, you might say, well, maybe the batteries are dead. What's another potential hypothesis? Maybe the light bulb is blown out, right? Again, let's get in the Wayback Machine before they were LED. And that would be even more likely. And so maybe the bulb is burned out. And that's supposed to be B, whoops. Sorry. Okay. So our observation is that our flashlight doesn't work. Why doesn't the flashlight work? Well, maybe the batteries are dead. Maybe the bulb is burned out. It's okay to have multiple hypotheses. In fact, it's actually stronger to have multiple hypotheses. The whole notion of what we call strong inference in science begins with having multiple working hypotheses that you sort of systematically eliminate with your experiment, right? So we have two solid hypotheses that we can test. If I want to test hypothesis number one, what might I do? Replace the batteries, right? So I can make a prediction if I replace the batteries, then flashlight will work. The experiment for this is really simple. I'm going to open up the flashlight and dump out the bad batteries, put in some new batteries, screw the flashlight back together and see if the light comes on. Okay. If I do that experiment and the light doesn't come on, and I want to instead test hypothesis number two, that the bulb is burned out, what do I have to do before I can begin to test that? We have to remove the bulb, but before I do that, put the old batteries back in, right? Because my original question is, why doesn't the flashlight work? I tested the batteries, and the batteries didn't make a difference. So I should put my original batteries back in before I do my next test, or else I'm complicating my two experiments, right? And I want to keep them independent. So I put the old batteries back in, and then I say, if I replace the bulb, then light will work, right? So I take off the top of the flashlight, pull out the bulb, put in a new bulb, put it back together, hit the switch, and the light comes on, right? When that happens, I say, oh, hypothesis number 2 is supported, right? The bulb was burned out. Do I have indisputable evidence that the bulb was burned out? No. Why not? Right. So I can say that my result supports my hypothesis, right? My hypothesis was the bulb was burned out. Replacing the bulb fixed the problem. My result supports my hypothesis. Unless I take that bulb and actually test it, look and see if the filament is still connected, or test it by applying energy to it outside of the flashlight. I don't know whether it was actually the bulb. Because like we just said, you could have easily had the bulb not making connection to the battery, and all this taking apart and putting back together of the flashlight you've done, you've now corrected the connection, right? So your data support your result, and that's good. And you say, okay, I have evidence that supports my hypothesis that the bulb was dead. But you can't say, I know with absolute certainty that it was the bulb. Right? You would want to actually check the bulb itself, just like you could put the batteries on a voltmeter and see what their output is. Right? You can do this simple experiment, but there's always an option to know a little bit more. Okay, so I like this example, even though it's getting a little dated, because we have the potential to see beyond the experiment we do. And frequently when we do experiments in science, we're like, aha, my results support my hypothesis. I know truth with a capital T. And it's not until 20 or 30 years later that we understand the system at a different level and are able to say, oh, actually, we only knew this small slice of truth, and now we have this additional information. So again, just keeping in the back of your mind that your results support your hypothesis. You had a positive outcome, but you would need to test that outcome again and find additional data to support it. We're not going to have a bunch of bum flashlights in the same condition, but in the kinds of things that we tend to be testing in biology, we'd be able to test that same system over and over again. Okay, so this gives us our scientific method, right? We've been working through most of this repeatedly these last 2 lectures. We start by observing, we go out and make an observation, and that observation leads us to a question. So we ask that question. Okay, frequently in your labs, I'm going to give you a research question. And so you'll start there. And from there, I'm going to expect you to develop a hypothesis. I like to just think of our hypothesis as an answer to the question. So why doesn't the flashlight work? The flashlight doesn't work because the bulb is burned out, right? Give me an answer. Tell me, it's good to tell you why you think that. You might say, well, most commonly, when my flashlight doesn't work, changing the bulb fixes it, or changing the batteries fixes it. Then you can make a prediction. If I change the bulb, then the flashlight will work, and I can test it. I design an experiment. I get a new bulb from the store, which is presumably in good condition. I put it into the flashlight. I see if the flashlight works. Okay. In that case, our result is really pretty clear. Either the flashlight turns on or it doesn't, right? In a lot of the experiments that you guys are going to do, you might get some numbers, right? And you might have a hard time knowing whether those numbers are different from each other or similar to each other. So you might even have to use some math to try to determine what the results actually are, okay? So you're going to analyze your results, and then you'll report those results. In lab, you'll put them in your lab report, right? In science as a whole, we report those results to other scientists, either at conferences, through poster sessions and through talks, or through the literature by publishing it in the peer-reviewed literature, right? And we always want to come back and repeat. You want to make sure that your experiment has been repeated many times and you continue to get the same outcome, right? Or compatible outcome. You can have a better idea of what's happening when you have a larger sample size, right? The more samples you take, you sort of are able to suss out what is the continuum of results from those samples. If you have one really odd result that sort of is an outlier and doesn't fit with all of the rest of the results, that can have a really big impact on a small sample size, but a less impact on a really big sample size. And so you're having more samples allows you to get closer to what reality is. And those repetitions allow you to know if the result that you saw was kind of accidental or if it's a pattern, something that's happening over and over again. When you get down to your analysis, regardless of whether or not your hypothesis is supported, you should report your results. In practice, right now in science, we have a real problem with only reporting positive results. If something doesn't go as we expected it to, we don't tell anybody. And our publication system actually prevents that information from going out in an easy way. But it's just as important to talk about your failures as it is your successes, right? Because you learn from those. They help you to shape and change your hypothesis for the next test you're going to do, right? So it's really important to think about what went wrong, why you didn't get what you expected. Was there something that you didn't understand about the system going on? That information is really valuable, and you should still share it with other scientists. If your results are not supported, you might come back and ask a new hypothesis, right? You might take what you learned from that experiment and use it to change the answer to your question. Try again. So let's try this with an example from the real world, from ecology. So here we have two individuals of a type of bird that's known as a widow bird. This individual over here is the male, and this individual over here is the female. So they're showing something we call sexual dimorphism, where the males and females look different. And so when scientists were describing this bird, they found it in the wild and they saw that these two individuals were clearly mating with each other and members of the same species. They said, well, our observation is that these males have really long tails, right? Their tails are considerably longer than the female tail. They have a different overall color, and it seems like maybe this tail would be cumbersome for flight. It might actually prevent them from being aerodynamic, right? So the observation is that males have really long tails, and the question is why? Like, why would males have such long tails? Especially when the female tails are a completely reasonable, sort of normal size that you would expect for a bird of that overall size. So what do you think? Do you have any answers to this question or hypothesis? Maybe the males have long tails because the females like long tails? Solid hypothesis. Does anybody have a counter hypothesis? Well, maybe the tail plays a role in nesting. Might have to do with how they make or protect their nest. It's a good hypothesis. Anybody else? Science as a whole, when confronted with this particular scenario, almost always goes with your hypothesis that males have long tails because females prefer to mate with long-tailed males. Generally speaking, in the animal world, males have to attract females to them. And so the females get to choose who to mate with because usually there's a surplus of males, we would say. There are more males in the population than are required to adequately mate with all of the females. And so the females sort of get their pick of the individuals out there. So there's a reason to have something that might make you more desirable to the female. So the hypothesis is that males have long tails because females prefer to mate with long-tailed males. And the prediction then is if females prefer long-tailed males, then males with artificially lengthened tails will attract more mates. So we're setting up an experiment here, right? We could make the tail even longer and see what happens. We could make the tail shorter and see if that individual with a shortened tail gets fewer matings, right? And this is where we do cruel things to male birds, because this is a real study that occurred, I think, in the mid-70s, if I remember correctly. We went out and sampled a population of male widow birds, and we took those individuals that we captured, and I keep saying we, I wasn't a part of this study, but science went out and captured these individuals and divided them into 4 groups, okay? The first group gets captured and handled, but the tail is not changed, okay? So once the male has been sort of processed in the same manner as all of the rest of the individuals, his tail is unmanipulated, and we release him and go out and follow his behavior. The second group gets their tail cut. But once they cut the tail, they re-glue the feathers right where they had been cut off of. So you sort of cut the tail and then glue all the feathers back together to where they had been originally. The third group gets their feathers cut to half the original length, and those feathers are taken away. And the fourth group receives the feathers from the third group. So this group gets a shortened tail, and we take the feathers from that individual and use them to artificially lengthen the tails of these individuals. So this is a control. We have 2 types of controls here. In this first control, the animal is just being handled, right? So we're trying to control for the capture, handling, processing piece. And then here, there's a second control just to make sure that there isn't something about having your tail cut and glued. Like maybe the glue has a smell and the ladies are like, I really don't like that smell, right? And so now all of a sudden, any individual who has glue has plummet in their mating. That isn't necessarily having anything to do with length, but that's an effect of glue, right? So this particular manipulation is to try to rule out the impact of cutting and gluing. And here, with these 2 controls in place, we can actually look at the impact of what we're really interested in, a shortened tail versus a lengthened tail. Okay, so we have those 4 groups. We have essentially 3 different tail lengths. We have normal, natural tail length, which is going to vary a little bit between individuals, but we measure that and accommodate that. And then we have sort of half tail and double tail. Okay, all individuals are released. They go out and set up their nesting territories. We give them a week to attract females and set up nests. These individuals tend to be polygamous, so one male will set up a nesting territory and several females will build a nest with him in that territory. So then, or several could. So then we go out and count the number of nests, right? If if there's no effect of tail length, we would expect every group to have the same number of nests, right? Maybe every male has 1 nest, as an example, right? But the results were that in both of our control groups, we had about 1 nest per male. In our shortened tail length, we had about half a nest per male. And in our doubled tail length, we had 2 nests per male. So those data are pretty strongly supporting our prediction, right? Female widow birds prefer to mate with long-tailed males. The longer the tail, the more nests that are formed. The shorter the tail, the fewer the nests, right? When you have an average of half a nest, that means that at least, approximately half of the individuals with a shortened tail aren't getting any nesting at all, right? Those females are over there doubling up on this long-tailed individual instead. So that's actually just an example from the literature, from biology. A very simple experiment, kind of mean to do that to those males, but it gives you an idea. Okay, what we're going to talk about, I jumped way far ahead. Here we go. I also want to just briefly mention that these sort of completely controlled experiments are somewhat rare. We can't always control all the environmental factors, especially when we're looking at experiments in nature, and also especially when we're looking at experiments in which humans are the subjects, right? So if you think about medical literature, right? Imagine we do a study on diet, right? And we're all supposed to go on a prescriptive diet and report back to our interviewer about what we ate that week, right? If you're on a particular diet that's supposed to be for managing diabetes or for weight loss, you're definitely not going to tell the interviewer about the cookie you had on Wednesday, right? Like, you're totally going to cheat, and you're also totally going to lie. Human subjects are notoriously tricky because they don't always do what they say they do. There's the stigma that smoking is bad for you, right? Are you a smoker? No, don't smoke, right? Lie on your intake interview, and then all of a sudden we put you in the wrong category when we compare results. So often what we end up doing are these comparative experiments. And so in a comparative experiment, we say, okay, we know that our experimental groups are going to vary. We can't control them perfectly. They're going to be different. Because of that, we're going to just try to measure the differences between the groups, and we're going to try to account for as many variables as possible. So if we wanted to look at lifespan in humans, we would look at things like sex of individuals, smoking history, dietary history, underlying genetic diseases, and we would make all of those things that we knew about that individual a variable we would try to compare between groups. And we would try to gather as much of that information as possible and then use statistical modeling to try to analyze what trends fall out, despite the fact that those individuals are variable. Okay. There's a lot of really sophisticated math that we're not going to cover in this class, but that you'll encounter as you move forward that helps us to sort of drop out some of these variables and elevate the thing that we're interested in, despite the fact that these groups vary a lot. So those studies can be done. We're going to keep things pretty controlled in here because we're going to be building really simple experiments with known materials in front of us. Okay, but know that these kinds of experiments exist and are successful and are still informative and give us good information as long as they're done really precisely. Okay. The last step in all of this, once you have done your experiment and you have your results and you've made sure that you feel pretty confident in your results, the last step should always be to communicate those results. Okay. You want to communicate them so that other scientists can repeat them and make sure that they get the same result. This happens all the time. Somebody performs an experiment, they get a result, they think, I understand how the world works. They publish their work and nobody else can get the same result, right? So then you have to figure out what happened. Why was that group able to get that result? Was it something very specific about the facility they used or the equipment they used? What was going on, right? This is how we share information with the rest of the world, both communicating with other scientists and also communicating with the general public, right? Generally speaking, scientific work goes through the process of what we call peer review. Now, when we say peer, who are your peers? What does that word mean? Other scientists in your field in this case, right? Those of you in this class, you are all peers because you are all taking Bio 1, right? You're at the same point in your biology career for the most part, right? So peer review is very much done by other people who are very knowledgeable about the thing you're studying. So I studied animal behavior. I studied foraging behavior in rodents. Somebody who studies biomedical physiology of rodents would not review my papers, right? Because they don't know anything about foraging behavior. Likewise, I know nothing about biomedical physiology. I wouldn't review their papers. Right? You have this small group of people who are also experts in your field, and they can tell whether or not you've done your experiment correctly. You've used the right analysis for the kind of data you collected. Did you draw the same conclusions they would have drawn given the same data, right? And so other scientists read and evaluate this work and weigh in on its value. Many papers don't get published because the peers are like, well, that's junk. That's not worth communicating to the general world. You didn't do your analysis correctly, You need to go back and use a different mathematical model. You need to go back and collect more data because your sample size is too small. There are lots of reasons that a paper might be rejected. If the peers generally agree that this seems solid and good, it moves forward. This is not infallible, right? Papers are sometimes retracted. Scientists are found to have cheated and manifested their data instead of collected their data, right? It happens, especially when people's jobs and careers are on the line. Some scientists fall to temptation and manufacture data that aren't real. Those things all happen. So this process isn't perfect, but it's another check to make sure that the information is strong before it moves forward. So generally speaking, only those papers that pass peer review go out to the published world. So in lab on Wednesday, we're going to look at some science information from the peer-reviewed literature and some science information from the general environment, from magazines and from news stories and from websites and that kind of information. And we'll sort of compare and look at it. But it's important to remember that really your ability to review that work is limited to the information that you're the audience of, right? Just like I can't review biomedical papers, you probably can't review foraging behavior papers. And so you probably can't, at this point in your career, review any primary literature because you don't understand the methodology well enough yet. And so you're going to be looking at things at another level. I mean, keep that in mind as we look at science information this week in lab. Okay. So that is the end of chapter 1. And what I want to do now is allow you guys a chance to review some of the questions that we've had in chapter 1. So I'm going